plot, examining the how long the patient survives. The red plot refers to the Black cohort, and the blue plot refers to the white cohort. There's really no statistical difference when we examine the two groups as a collective. When we look at the more advanced stage, where the patient typically is older, over 60 years, a difference begins to emerge, even if it's not statistically significant. We see that the Black cohort tends to die off after about 1,000 days, whereas the white cohort survives much longer. So examining the data and looking at the patients that are in there, I've been particularly curious. The study from Jos, Nigeria, identified the onset of breast cancer of the aggressive kind in the Nigerian woman to be much younger. The average age, the median age being about 43. Whereas if we look in Norfolk in the United Kingdom, a similar cohort, the average age of onset was about 64. So looking at the different stages, we find that overall there are fewer samples from Black patients than there are from white patients. If we look at all ages at onset of diagnosis, we have the same pattern. There are fewer Black patients than there are white patients. But making do with what we have, if we look at all the ages of all the patients pooled together, there is no statistical difference in terms of prognosis or survival. However, when we look at, this is stage 2, right? Stage 2 breast cancer. When we look at those who are 50 years old or younger, 50 years old or younger at diagnosis, we find that the Black patients die sooner, and that's highly statistically significant. The challenge, though, is that there are only so few Black patients for whom survivor data was gathered. All right, so then we begin to probe this even further. If we look at stage 3 patients who are older, stage 3 and 4 who are older, we find that difference still remains. Over age 60, if you compare the Black patients with the white patients, it's very statistically significant. So ordinarily, we've been interested in transcriptional regulatory networks. We've been looking at other domains, but cancer has become increasingly interesting. I know the chair, Emmanuel, has done quite a bit of work looking at regulatory networks of the Bayesian kind. But we have also used other approaches. Computationally speaking, we've been using transcriptional regulatory networks of a variety. In this chart, we're looking at high throughput gene expression data representing different genomic perturbations. There can be perturbations due to drug exposure, or else due to genetic perturbations, whether they're overexpressed or knocked down. These are put together to generate a compendium. In this particular study, they had 445 arrays, 445 samples across 189 conditions. And then to this compendium, appropriately processed, is applied as a learning algorithm. Very often there's very little supervision, except in some cases where we identify which ones are likely regulators by virtue of whether they are transcription factors or else other regulators in other formats. So based on this learning algorithm, machine learning algorithm, we can infer regulatory relationships, networks of regulatory relationships. So these nodes each represent genes. There are regulatory nodes and target nodes. And these give us a set of hypotheses which can then be validated. Validated either by way of databases, which contain experimentally determined regulatory relationships, or else validated by way of the literature or experiments. And the idea is to discover novel patterns and novel relationships that can shed light on the particular disease. So we have set out to use transcriptional regulatory algorithms to explore lung disease, to explore neurodegenerative disorders, and more recently to explore cancers, particularly breast cancers. And lately we've been working with a group called PanCan, and they are interested in pancreatic cancer. Okay, so there are a number of different approaches. And in this particular approach, we use mutual information, where we have discrete variables, X and Y, X representing a regulator gene, Y representing a target gene, individual expression values represented by XY, XI, and YJ. This is the formula for computing mutual information. These computations enable us to determine whether any two nodes are related. If in fact there's a relationship, regulatory relationship between X and Y, then that relationship should be evident through the different perturbations across the different samples that are presented. So mutual information is computed. The relationship can be linear or otherwise not linear. It can be positive or otherwise negative. But we use these as tools to probe regulatory relationships that could exist between genes. So this is an example. Here looking at the two groups, Black Africans, Black or African Americans, and we're looking at genes that are suppressed within them, represented by the white nodes. This is one such hub, and it, HMGA1, high mobility group AT-HOOK1, happens to be one such regulator of note. We've examined several different regulatory networks. In this particular one, we are using an approach called Arachne, which is one of the inference algorithms. And here we identify relationships that are of interest. Some of these have to do with ubiquitin conjugate bases, oncogenic targets, and others. The nodes that are green have suppressed expression. The nodes that are white have elevated expression. And then we begin to explore them for etiological or mechanistic insights. Pushing this a little further, we go from gene expression patterns to the inference of protein expression and protein activity levels. And there are established computational approaches. So we go from gene expression profiles, high throughput, large compendia. From those, we determine gene expression signatures. And we use the transcriptional regulatory network inferred by one or other algorithm. In this case, we use Arachne. And we make our way towards inferring protein activity profiles. Right. So we used both of these in this work. So when we look at stage 3, all ages, patients of all ages, we look at stage 3. These are the top 20 master regulators. Each of these represents a regulon, so to speak, a group of genes that are regulated similarly. And at the hub, we have the master regulator. Each of these bars here represents a gene that is a target of HMGA1. And we find that there's an enrichment of the HMGA1 regulon among the Black or African American cohort. HMGA1 has low expression in non-malignant tissue, but it tends to have elevated expression where there's cancer, and particularly where it's aggressive. There are other nodes, master regulators here that are of interest, but for want of time, we'll dwell on HMGA1. I have talked about TERT in the past, being a key player in an immortalization gene set. It too has differential activity between the two cohorts. So ubiquitin tagging basically leads to proteasomal degradation. So first there's the activation, then there's the E2 ubiquitin conjugase, then it gets transferred to E3 ubiquitin ligase. There's the ligation that leads to ubiquitination of the target. Ubiquitination leads to polyubiquitination, in particular, leads to proteasomal degradation. So it becomes a negative regulator of the protein target. Countering that, we have deubiquitination that essentially reverses ubiquitination. So between the forces of deubiquitination and ubiquitination, we can exert a regulatory influence on what happens in the process. There are a variety of intrinsic molecular subtypes of breast cancer. There's the luminal A and luminal B, which have expression of hormonal receptors, namely estrogen and progesterone. But the luminal A has no growth factor, human epithelial receptor being expressed. And then luminal B is similar, except it does have the HER2 expression. But then of interest is the triple negative breast cancer. Triple negative breast cancer. Triple negative breast cancer tends to be bad news. It doesn't have those three receptors: estrogen receptor, progesterone receptor, or even the growth factor receptor HER2. Prognosis for triple negative breast cancer is particularly bad. When we examine all patients, here we're looking at immunohistochemistry. We find that the triple negative breast cancer, looking at all the patients available for whom the data was available, about 35%, it occurs at a rate of about 35%. But then if we look only at Black or African American patients, the rate is much higher. It occurs at a rate of about 56%. It is also somewhat higher in the Asian population compared to the white population. So as studying triple negative breast cancer can give us a window as to what is happening more among Black patients, because the incidence rate for triple negative breast cancer is much higher. Now, if we look at the non-triple negative breast cancer, we find that occurs the most among white patients. The incidence of non-triple negative breast cancer is lower among Black patients. We find when we do gene set enrichment analysis, we find that the genes that are involved in ubiquitination or else sumoylation has to do with small ubiquitin-like modifier molecules. So we have SUMO1, and we have an array of ubiquitin ligases and conjugases. HMGA1 itself is one of such. Remember, we have identified HMGA1 as a top master regulator at stage 3. We find that genes that are involved in whose products are involved in sumoylation, ubiquitination, are expressed at higher levels. The red here represents higher levels among the triple negative breast cancer cases at all ages. And then if we flip it and look at the targets of ubiquitination, those targets at which the whole ubiquitination and proteasomal degradation is aimed, we find that those genes have suppressed expression in triple negative breast cancer. So the blue here represents decreased expression. We find that those genes are suppressed in triple negative breast cancer. So what this tells us, and this is looking at the patients as they're presented. Some have triple negative breast cancer based on the immunohistochemistry. Others have non-triple negative breast cancer. Among those patients who have triple negative breast cancer, sumoylation, ubiquitination genes are elevated expression, whereas the target genes have suppressed expression, which is what you would expect given the nature of ubiquitin tagging followed by proteasomal degradation. Then taking our attention away from the Cancer Genome Atlas, if we looked at an independent gene set, here, this comes from the Gene Expression Omnibus, GSE gene set, data set GSE 47994. We find a similar pattern. Ubiquitination genes, sumoylation genes have elevated expression in the triple negative breast cancer cohort relative to the non-triple negative breast cancer cohort. We have multiple rows for some of these genes because these are microarrays, and sometimes you have multiple probe sets representing the same gene. And similarly, the targets of the ubiquitination simulation process have suppressed expression, just as we saw with the triple negative breast cancer, with the Cancer Genome Atlas data set. Then if we focus on those younger patients in this independent gene data set, we find the same pattern. Patients who are 50 years old or younger. We find that simulation ubiquitination genes have elevated expression relative to, in triple negative breast cancer relative to non-triple negative breast cancer. And then the targets are also expressed, just like we saw when we did all ages. All right. If we look at mutations, we also examine somatic mutations. And we find that mutations in PIK3CA, the catalytic subunit of PI3 kinase, those mutations tend to be protective. And we find those mutations occurring more frequently in the non-triple negative breast cancer samples. Incidentally, we find those mutations, the PI3K3CA, to also be occurring at a higher frequency among the white population compared to the Black population. So HMGA1 remains a master regulator, driving the difference also between triple negative breast cancer and human epithelial receptor 2 negative samples, right? So this remains an obvious target, and there's much in the literature across a variety of cancers affirming that HMGA1 is indeed a master regulator of the progression and metastasis in breast cancer. Now, looking at triple negative and non-triple negative samples, we see again HMGA1, and we have done my former master's student, Rosa Lesongo. She's now in the Congo lecturing. We find that she actually established, validated the link between HMGA1 and PPAR-D. There are other players in the process that occur on this chart. This here represents a gene set enrichment plot, an enrichment plot. Here, if we look at the genes as they are expressed in non-triple negative breast cancer and triple negative, if we rank their expression from the most highly expressed to the least expressed, we find that a collection of genes whose products are engaged in protein-protein interaction across a variety of pluripotent cells, ranging from some of the different kinds that Professor Jay talked about yesterday. Across those varieties of cells, there's this collection of protein-protein interaction that is common to them. And this is together labeled here as plurinet. And we find that there's a gathering of those genes at the bottom of the ranked list. And the way this works out, there's this Kolmogorov-Smirnov kind of computation, where if we encounter a gene on the ranked list that is not in our plurinet gene set, there's a penalty. And if we encounter a gene that is in our plurinet set, there's an addition. And we find a rise in the plot here, indicating an enrichment within the triple negative breast cancer. So if we look at the plurinet gene set, the leading edge genes, those genes that result in the rise, they gather here in the bottom right, red here indicating high expression. So those genes, we've listed them here. Those genes are expressed highly in the triple negative breast cancer cohort relative to the non-triple negative breast cancer cohort. Here we see some of the same suspects, the simulation and HMGA1, HMGB1, MYC, and others that occur in the leading edge. And if we just separate out the leading edge genes, we find that there's elevated expression in triple negative breast cancer relative to non-triple negative breast cancer. Then there's stemness. There's stemness. Yesterday, Professor Jay talked about the Yamanaka factors. This is a collection of genes that are involved in stemness. And we find that they too occur at elevated levels in the triple negative breast cancer cohort relative to the non-triple negative breast cancer cohort. All right, so now we turn our attention briefly to some of the mutations that occur. And here we are looking only at stage 2 and younger patients, patients who are 50 years old or younger and who are at stage 2 breast cancer. We find that the protective PIK3CA mutation occurs more frequently in the white cohort than it does in the black cohort. And the HUWE1, which is a ubiquitin ligase, we find that it occurs, mutations among it, among Black patients are found more frequently than they are among the white cohort. So we're interested in what this means. Does the fact that these two mutations occur more frequently in the different cohorts, does it have any direct consequence on the survival? So here we see the PIK3CA mutation occurs very often in the white cohort, and they are all missense mutations, very low occurrence rate in the black cohort. Whereas with the black cohort, there are fewer patients, but we find a 17% occurrence in the black cohort, whereas in the white cohort, it's only a 3% occurrence. And for the most part, these are missense mutations. Do these mutations alone account for the difference in survival? That would appear not to be the case. So looking at the Kaplan-Meier plot, we don't see any statistical difference in the survival between the Black patients and the white patients. Here we only had 5 patients with the, this is looking at all patients who happen to have the mutations. There were 5 who had the mutation and for HUWE1, and 22 with the mutations for PIK3CA, and there was no difference. And this is just looking at the mutations alone and not looking at age, not looking at race or anything else. But what this tells us is that the mutations by themselves alone do not say much. But here, looking at the different intrinsic molecular subtypes of the breast cancer, we find that if we look at all patients with HUWE1 mutation, the vast majority of those patients happen to have basal-like breast cancer. They happen to have basal-like breast cancer. Most basal-like breast cancers are triple negative. On the other hand, if we look at the patients who happen to have the PIK3CA mutations, very few are basal-like. By far, most are luminal B. Most are luminal B. All right. So what this tells us is that the patients with HUWE1 mutation tend to be basal-like, or we assume here, because most basal-likes are triple negative or triple negative in nature, whereas PIK3CA mutations occur less frequently with the basal-like, and more likely to be luminal B. Then we look at gene expression. And some of the interesting trends occur here. Genes that are involved in ubiquitination, SUMOylation, are expressed differently between those patients who are HUWE1 mutants and those patients who are PIK3CA patients, 50 years or younger at diagnosis. Of particular interest here is PTEN. PTEN is a tumor suppressor. PTEN is a tumor suppressor, and PTEN loss or suppression of PTEN expression is generally bad news. It's associated with aggression of breast cancer. And here we find the red here represents suppressed expression. We find that the PTEN expression is severely suppressed in those patients who have HUWE1 mutations, compared to those patients who have PIK, excuse me, compared to those patients who have PIK3CA mutations. There are a number of studies we looked at. We looked at protein expression, but time will not allow us to get into that. HUWE1 is also known as ARFBP1, and it is inhibited by ARF. It's a ubiquitin ligase. It tags p53 and makes for p53 degradation. But when HUWE1 is suppressed, then p53 is more stable. There's more apoptosis, and there's less replication and aggression, p53 being itself a tumor suppressor. So inhibiting HUWE1 stabilizes p53 and and tends to be protective. Again, it's HUW1 is a ubiquitin ligase targeting MYC and making it targeted for degradation, but it gets inhibited by ARF. These mutations here, looking at Black patients 50 years older or younger, these HUW1 mutations co-occur with other mutations, FBXW7. Actually, it co-occurs with TTN, and the TTN mutations co-occur with FBXW7 and HYDIN. So all of these give us insights. We find different master regulators at different stages. HES4 and PQBP1 seem to occur at all the stages we've looked at, but here we see it in stage 1. When we go on to stage 2, 50 years old or younger, we see the same HES4 and PQBP1 along with others. It is only when we get to stage 3 that we see HMGA1 occurring among the top. So in sum, we find a set of molecular players that are relevant to the differences driving the fact that the breast cancer in Black people tends to be more aggressive, tends to occur at a younger age on average than the white patients. HMGA1 representing modulation by way of ubiquitination. TERT, which we didn't talk much about, representing an immortalization gene signature. These are all important players in the process. All right, so I know we've talked about many molecules here, but this is a convenient place to stop and invite questions.
Thank you very much, George, for this presentation. Like he said, we welcome questions from the audience virtually. You can type your question in the Q&A box or in the chat box, or you can raise your hand and we will see and call you. All right, I have one hand raised. Kwesi Nyamfo, you can speak now.
Thank you very much. Yes, breast cancer is very serious in Africa. And I want George to tell us whether if they are working with African Americans, they are assuming the same results for Africans, because there seems to be a change between us. So that's the first one. And then second one, which one in particular, like HMGA1, TET, PIK, there are a lot of regulators that he's mentioned. Ghana hasn't got the time and money to go into all this. So for a Ghanaian researching into breast cancer, molecular aspects of breast cancer, which one should be our target to identify? Because before then, we all thought BRCA1, BRCA2 was the target, but now which one should be our target that we should follow, since we don't have the time and money? Thank you.
Thank you very much. So yes, BRCA1 and BRCA2 remain important. In fact, one of the things I forgot to mention, HUWE1 tags BRCA1 for proteasomal degradation, right? So HUWE1 tags BRCA1, and BRCA1, as we all know, is a tumor suppressor. and when there are mutations in BRCA1, and then the person becomes prone. In fact, mutations, BRCA1 mutations are a marker that a person is likely to get to encounter breast cancer down the road. But we find that BRCA1 itself, the founder mutations in BRCA1 occur most commonly among certain ethnicities than others. If you are an Ashkenazi Jew, the BRCA1 mutations are particularly interesting. So those mutations that we generalize as being marker mutations may be more relevant to other ethnicities than they are to Black or Africans. The other question you asked was, what does one focus on? So these data are available for use. They can be mined by anybody anywhere. It's not, there are some of them that are more privileged and for which special permission, we have to go through a process to have access to, but otherwise these are generally available. People use them across the world. So which one to focus on depends on one's really, there are all of these things that I've mentioned are possible hypotheses. And you'll find this as you probe the literature that one or other may pique your interest, and that could be your cue to pick it up and run with it. I mean, there are things that can be done to confirm. There are things that are in the literature that might make you want to take one particular hypothesis as opposed to the other, but all these are linked.
And are you assuming African Americans and Africans are the same?
Not necessarily, but anybody who identifies as Black. So some of these designations are, in all fairness, problematic because genetically, if we say somebody is Asian, they could be Indian, they could be Japanese, they could be Qatari. So some of these designations are not specific enough. But this is an American-funded project. And so if someone comes and says they are Black, they label, their notation in the data is that this person is Black or African-American. So you note that there's that designation, Black or African-American. So there's not an effort here to tease them apart. But apart from that, there have been studies, I mentioned two of them that make the same basic findings, that the breast cancer among Black people is more aggressive than the kind among white people, particularly at the younger age.
Kwesi, I hope you are okay.
Yeah, I'll see him.
All right, so you put your hands down. I saw, okay, wait, so you have hands down. I saw two hands. I think the guy has put his hands down. Let me call Dr. Kusi.
There are two that have been typed in.
Yeah, let's handle the hands raised first.
Okay.
Yes.
Okay, so indeed, the question I had is what has been answered just now, whether there is a difference or between African Americans and typically Africans. I think, is it possible to answer that question to source for the information from typically African populations for comparison with African Americans, or is it that not much of this has been done on the African continent for such data to be available?
Yeah, I think there has to be a deliberate effort to gather such data. But you know, when you talk about Africans, if I had a colleague who came from Algeria, she's African.
Mm-hmm.
And I am African, and I have had a student, a white student from South Africa, and he would tell you that he's African. So purity, ethnic purity is not something that we can claim here.
I think
within the group of African Americans, you have those. My children have pure African genes, right?
Yeah.
They will claim to be African American.
Yeah. Yeah. So for me,
African Americans that have more of a mixture across the gender.
For me, I'm also looking at environmental factors. So even if the genetics is the same, is the environment playing a role? So maybe that is wrong terminology. It's still African American and typically African, but for persons who have lived most of their lives in this environment compared with those who have lived most of their lives in a different environment, for example.
Yeah, that's a study that will be worth a while because there are so many things. There was a time when I wouldn't need to take malaria prophylaxis when I'm in Accra. But I have learned that when I'm there, I'm not as resistant to mosquito bites as I used to be, years ago. So some of the findings that will be made here may not necessarily be applicable for environmental or non-genetic reasons.
Okay, thank you.
Yeah.
All right, so let me pick Bernard Asantis first before Prof. James Ajayi. So Bernard. All right, Bernard is muted. I don't know whether he's speaking out or not. But whilst we wait for Bernard, Prof, can you attend to the question in the chat box?
Okay. Bernard is someone is trying to speak.
Yeah, am I on now?
Yeah, you are on. All right.
Okay. Thank you so much for the presentation. Doug, and I would want to find out the factors that enhances the initiation of the deubiquitination. So that's what I want to find out. The factors that enhances the initiation of the ubiquitination. Yeah.
Okay, so there are a variety of where do I have that? So ubiquitination, this is something I took from the literature.
Okay.
There are a variety of E2 conjugates and ubiquitin E2 conjugates enzymes, E3 ligation enzymes, and the expressions levels and activities of these enzymes will have everything to do with the tagging of the target with ubiquitin molecules, which then makes them amenable to degradation by the proteasomal complex.
Okay. All right. Thank you so much.
Yeah.
All right. So you can attend to the question in the chat.
Okay. So I think Adam has a question. Let me see. So the data sources. So these are looking at breast. So those are the samples of interest. So looking at the breast tumors and extracting protein, doing the RNA-seq, doing the DNA methylation. We have a whole other set of data that examines the DNA methylation patterns and how that impacts gene expression and the regulation of the gene expression within the two cohorts. But these are very specific to the organ.
So thank you very much, Prof, for responding to these questions. Let me call Prof. Ajayi to also ask his question. Hello, James.
Yes. Can you hear me now? Yes, I'm unmuted. Yeah.
Yeah.
Yeah. So what I was going to say was, I think it was very nice showing how you carry out this meta-analysis to identify genes associated with the regulation or prognosis of various cancers. Is there a similar database where you're not looking at just the expression, the gene expression, but rather mutations?
Yeah.
So how do you correlate this to a mutation? So for instance, the p53, we know that it's a mutation, mutated p53, etc. So how do you correlate or reconcile the gene expression levels and the mutations of these genes and the downstream induction of the cancer?
Yeah, so thank you very much, James. So there are a variety of data types that in this particular resource are available. So there's protein data, there's somatic mutation data, copy number variation data, yeah, so what we're trying to do is to look at all of those. We do them in isolation, we do them together. So for instance, here, this is an attempt to look at those patients who have mutations, the specific mutations of interest, and looking at the gene expression, the expression of particular genes of interest for those patients with the mutations. So we did the mutation analysis separately, and then here we're looking at the RNA-seq data separately, and then putting them together. So the resource lends itself to a variety of analytic approaches, but we just have to be creative and in exploring what is available. But besides the Cancer Genome Atlas, there are other data sets that are more targeted. Like I mentioned, I'm working with a group called PanCan, and they are specifically focused on pancreatic cancer. And they have a variety of data types, including treatments, when treatment was started, what the outcome was, whether the patient went through with the treatment, and all of that. So there's a proliferation of these data resources, and they present an opportunity for us all.
Okay, thank you, Prof. Akwamensa, for this insightful presentation. I think the discussions can continue through the chats or contact him for by email and continue the conversation. Our time is up, and I'll hand over to Dr. Asamoah Kosi to continue with the next session. Thank you very much.
Okay, so thank you very much, Dr. Adabo. I think this is going as planned. So good afternoon, everyone, and welcome to the next session. So this is a session on bioinformatics, food safety, and chemistry. 2 sessions. I'll take the first one, and Dr. Anania Akolo will take the second one. So without wasting much time, we have 4 presenters, and I'll introduce each one as they prepare to give their talk. So the first to present in this session is happens to be the moderator for the just ended session. That's Dr. Emmanuel Adabo. He's from the School of Technology at GIMPA in Accra, and he'll be speaking to us on assessing antigenic similarity between influenza A viruses using a computer-based model. So I will invite Dr. Adabo to share his screen and get the presentation underway.
All right, thank you. Thank you very much, Dr. Kosi, for introducing me in a nice fashion. This afternoon, Emmanuel, like you said, I'm talking about assessing antigenic similarity between influenza viruses using computer-based model. My focus is on influenza because influenza A, H3N2, because of the abundant genetic data and its epidemiological importance. We know about influenza around the world that it causes close to 3 to 5 million severe cases, resulting in 350 to 500,000 deaths globally. So this research is very important, and we will assist in the surveillance with this particular work that I'm presenting on. I'll first talk about background, and then a problem, state my objectives and the methods that I'm using. It's a computational approach, so I'll show that, and then show some results, conclusion, references, and then provide some acknowledgements. It's a global effort of surveilling influenza evolutions around the world. And the WHO has established surveillance centers around the world, and their work is to monitor circulating influenza strains and then analyze antigenic relatedness between circulating and vaccine strains. That's what they do. Now, from these two studies, they will normally use what they find to review and recommend influenza vaccine strains. Now, the revisions of influenza vaccines are very necessary because of the frequent changes in influenza viruses. Therefore, they do this annually and make recommendations for vaccine design. Now, these recommendations are usually based on accurate assessment of antigenic similarity. Now, antigenic similarity will consider how to characterize the current circulating influenza strains, see whether it matches with previous strains or is a novel strain. And then based on that, they will recommend appropriate composition for vaccine design in future and for future strains. But there are issues. The characterization and the determination of antigenic similarity have been based on what we call hemagglutinin inhibition titers. However, the use of these titers have been criticized in the sense that it's not only composed of things that can determine antigenic variants, but also it has some non-antigenic non-antigenic particles involved. And so a more suitable approach could be an approach that will be devoid of non-antigenic variables. And so based on this, it was proposed that let's switch to the use of amino acid sequences. However, amino acid sequences, the models that incorporate these amino acid sequences are less exhaustive in terms of incorporating all the features or the parameters that accompanies antigenic variants. The importance of the use of amino acid sequences are that it's actually related or highly connected with the antigenic properties of strains. That's why it is recommended. But the problem has been that the use of it prevents people from using all the properties that you would expect in antigenic variants. Now, with this problem, it also affects the clustering of strains in that we will have for some season, some mismatches. Now, these mismatches may be due to improper characterization of strains. So what I intend to do in this presentation is to develop a computer-based model using distinguishing features of antigenic variants to analyze antigenic similarity among influenza strains. Now, the purpose of this is to ensure that we have a platform that incorporates all or most of the antigenic properties of accompanying strains in one place or in one model. And then after that, I will examine the relatedness of the model to influenza vaccine effectiveness. Now, this is how I proceed. First, I collected influenza H3N2 data from the protein database of the NCBI. And then I obtained the cluster information about these strains from 1968 to 2016. And then from knowing the cluster information will enable you to determine which strains are similar and which strains are not similar. Now we assume that, or it is confirmed that strains that belong to the same clusters are similar, and those that in different clusters are dissimilar. Now with this information so obtained, we go ahead to obtain amino acid sequences from NCPI, and then perform multiple sequence alignment for the data so that we can have equal length and extract the necessary information from the sequence data. Now, the information we extract are such that already we know from earlier studies in influenza that there are 5 designated antigenic sites and a receptor binding sites, which are very crucial for the escape of influenza viruses from antibodies. And so what we did was that we located the changes in those sites that accompany antigenic variant because they are known to cause antigenic variation. Now, with this data obtained from the data itself, using an R code, we produce a model, which is actually a logistic model, which I show in this particular slide. Now, once we develop a model based on this data, the model is evaluated and then compared with how it links to vaccine effectiveness. The evaluation will be based on what we call area under ROC curves, sensitivity, precision, specificity, and then accuracy. Now, the details of these are presented here. When we say accuracy is the sum of TP plus TN, all on TP plus TN plus FP plus FN. Now, TP is called true positive. Now, a true positive is an instance where we truly predict that 2 strains are similar. And then true negative is an instance where we truly predict that 2 distinct strains are strains which are distinct are actually distinct. And then we have the other parameter, which is FN, is false negative, which is the case where we fail to recognize distinct strains. And then false positive is where we incorrectly say that strains are similar, but in actual fact, they are not similar. Now, the same properties are used to define the precision. And then from the precision, I can talk about the area and the ROC curve. ROC is receiver operating characteristic curve. And this is a curve that plots what we call sensitivity against the false positive rate. Here, sensitivity is given by the expression that I have shown. Now, the area and the ROC curve shows the discrimination power of any computer model because it gives an indication of how accurate the model is. Now, all that we will do in this presentation is to present everything in a computer form. So I have here a computer implementation of whatever processes or methods that are presented, and they are implemented in R version 3.6.3. The performance was assessed using a 7-fold cross validation. So let's talk about some of the results that we had. This is a plot which represent the area under the ROC curves. Now the curve in blue denotes the ROC curve for the model we are referring to. And then the plot in gray, the straight line, is a random model. Now, how we measure accuracy is that your model should have ROC curve above the random model, which is the gray line. And here, my model is above it, and so it is satisfied. Now, the good thing about the model is that it is giving me an area of more than 95%. And you know, the good thing about this is that when you compare other existing models that can do similar tasks, they only reported area under ROC that did not exceed 90%. Now, besides the ROC, other performance metric also proves that the model was a bit accurate in the sense that the accuracy that was measured was 94%, and precision was 76%, specificity of 97%, and sensitivity of 68%. Now, above this, we also check the diagnostic odds ratio, which is expected to be above 1. And we had something around the 83, which is better than what you would expect in what has been done in other works, right? Now, the good thing about this is that there are other methods that are based on naive Bayes or multiple regressions. They all are able to solve such kind of problem, but the differences are that whilst we achieve high levels of accuracy, having 94%, others are only able to achieve 70%. For instance, a recent study by De Gucht and colleagues achieved accuracy of only 74%. and then that for others, then that for the precision was a bit higher, getting around 80%. And the same thing can be said of the specificity and then the sensitivity. The difference here is that we do not consider some of what they talk about. For instance, they did not talk about diagnostic odds ratio, but we are talking about it. And then in addition, the previous study did not consider the receptor binding sites that we are now considering in this particular study. So that could probably account for difference, but in all, these numbers shows how accurate the model is. Now, in addition to that, I said there are 5 designated antigenic sites. Now, these sites are designated A, B, C, D, and E. Now, we had to test the coefficients of the model we were presenting, and we realized that indeed all the parameters that we considered for this particular model for prediction were significant. When you say it's significant, it means that they were actually determinants of antigenic variants. Now they are determinant, and so that is acceptable because of the p-values, which are all less than 5%. Another thing we can also see is that when you consider antigenic sites A and B, they have the highest magnitude compared to antigenic site D and E. The significance of this is that previous studies have shown that antigenic variants that are likely to cause the next epidemic or pandemic will have changes in the antigenic site A and then site B. And so it is major driver. These sites are considered as major drivers for future variants. Not only that, the signs that we see by the antigenic site A and site B denotes that if you have any changes in antigenic site A and site B, then your strains will be more dissimilar. That is, they will be distinct. And this shows that if you are going to, or if there is going to be changes that may lead to seriously variant viruses that can cause epidemic, then we have to account for changes in A and B. However, when you consider changes in C, D, and E, although they are significant, changes there do not lead to increased antigenic dissimilarity. And the good thing about these results are that a recent study by Coel et al. In 2015 or 2014, they revealed that indeed changes in C, D, and E do not really cause any major antigenic changes in variants. However, changes in A and B cause these variants that accompany influenza epidemics. And so it shows that these are really significant. And of course, we had the same report for the receptor binding sites, which also indicated that changes around these sites will actually lead to antigenic variants, which are of significant epidemiological significance. The constant here denotes other variables that may also be responsible for the changes which were not accounted for in the model. And then besides that, we also had to ensure that this model is linked to influenza vaccine effectiveness, because that is the main purpose for this modeling. And so we ensured that our model was actually compared with the current data that we had. Now, the current data, which was from 1968 to 2016, we were able to get influenza vaccine effectiveness for these seasons in which influenza A, H3, and 2 were dominant. And using that data, we realized that the model was very correlated to the influenza vaccine effectiveness, which was 0.76 with this p-value. Now, what it means is that variations in effectiveness, variation in vaccine effectiveness, okay, can be explained by 58% of the model, can be explained by 58%, which indicates that this model is significant in the sense that we already know that there are other factors that could also account for influenza vaccine effectiveness, which include the immune status of vaccinated individuals and also age of individuals. So what I have done now is I've been able to show a computer-based model that could predict antigenic similarity between strains using these results to prove that. And then also the model incorporates the features which accompany antigenic variants, which unlike other studies is not available. And then also the model accounts for variations in influenza vaccine effectiveness, which has been shown. We believe that this work will complement global efforts when it is applied. Now, I'm saying we believe that because I have already presented this to Hele Leon, and it has gone through, I think, 3 stages of revisions, and that confirms for all the reviewers are confirming the relevance of this particular results or this particular study. But the issue that has been raised, which I'm considering for the next 2 weeks, is that the data on vaccine effectiveness should actually cover up to 2021. And it has been difficult getting this data, but I'm hoping that I'll get vaccine effectiveness data in the next 2 weeks and provide the necessary responses to the reviewers. So these are the references. The Indiforms paper describes the problems that are attached to the use of hemagglutination inhibition titers. And
okay, Dr. Rao, so I think we will look at the screen. You are like almost 2 minutes over.
All right.
Yeah, let me say thank you very much. As has been the tradition, if anyone has any question, please hold on, write it down, or type it in the chat box or in the Q&A session. And then at the end of all the 4 presentations, we will come back and take the questions. So thank you very much, Emmanuel, for the presentation. And without wasting much time, I'll invite the second speaker, Bernard Asante. He's with the Department of Chemistry at KNUST, and he will be speaking on anti-helminthic, anti-inflammatory, antioxidant, and antimicrobial activities and FTIR analysis of Vernonia camporum stemback. So Bernard, if you are on, please share your screen and let's get the presentation underway.
Okay, thank you so much. Good afternoon, everyone. I'm Bernard Asante, and I'm presenting on in vitro anti-helminthic, anti-inflammatory, antioxidant, and antimicrobial activities and FTIR analysis of Vernonia camporum stemback. And this work was conducted under the supervision of Dr. Clementa Seakoto and Professor Ekwesi Echampo. So lipid peroxidation reaction is an important deteriorate reaction.
Excuse me, we can neither hear you nor see your presentation.
Really? I've shared the screen too.
Yeah, Bernard, we can't see.
I've shared the screen.
Ekwesi, can you help?
Please, can you see now?
Yeah, it's coming.
Showing, yes. Okay.
Okay.
So go into presentation mode. Yeah. Okay.
Please, am I okay now?
Yes.
Yes.
Okay, so
do you have video? Oh, it's not an option. Okay, that's fine.
Please,
can you turn your camera on?
My camera is on, but I don't know why.
Okay, you go ahead. That's fine. You go ahead.
Okay, so I'm Bernard Asante, and I'm presenting on in vitro anti-helminthic, anti-inflammatory, antioxidant, and antimicrobial activities and FTIR analysis of Vernonia camporum stemback. This research was conducted under the supervision of Dr. Clementa Seakoto and Professor Ekwesi Echampo. Lipid peroxidation reaction is one of the important deteriorate reaction in human metabolism and food processing and storage. It has really caused detrimental health problems such as arteriosclerosis, carcinogenesis, mutagenesis, aging, and oxidative damage diseases. It is also reported to affect food quality and safety. So imbalance between antioxidant protection of the human system by exogenous factors and endogenous factors has led to the production of free radicals and non-radical species. And these species, which include hydroxyl and hydrogen peroxides, are known to enhance lipid peroxidation in the metabolic process of human beings and As a result, it's causing the disease mentioned above. Also, it is known worldwide that the predominant cause of death is infectious diseases. And WHO reported in April 2019 that about 700,000 people die each year due to drug-resistant diseases, predominantly infectious diseases. Also, Center for Disease Prevention and Control reported in December 2019 that 2.8 million antibiotic resistance infections occur in the United States each year, and more than 35,000 people die as a result. Drug-resistant diseases could cause 10 million deaths each year by 2050, and this was estimated by WHO in April 2019. Also, 24% of the world population, which is equivalent to 1.5 billion people, are known to be affected with soil-transmitted helminth infections. Inflammation is a normal body mechanism in terms of the body responding to tissue injury, and it involves a cascade of biochemical reactions. However, if inflammation is not treated, it leads to various diseases like vasomotor rhinorrhea, rheumatoid arthritis, arteriosclerosis, and many other diseases. So this is a serious challenge globally, and as scientists, we must be able to contribute our quota in solving these problems. Otherwise, in the next coming years or the near future, human beings are going to be endangered. And therefore, in contributing our quota, we realize that our natural products have contributed in various arenas in research like bioorganic chemistry, organic synthesis, phytochemistry, medicinal chemistry, natural product chemistry, pharmacy, and many others. So we decided to look into getting a solution from natural products, and we realized that 47% of drugs that are reported or that are used worldwide are either obtained from plant or are enhanced by plant, and 23% from fungi, 30% from bacteria, and 5% from other sources. And this was reported by Patridge et al. In 2016. Therefore, we decided to look more into plants in getting a solution for this. And from our review, we realized that 75,000 species of higher plants exist on Earth. Out of these 75,000 species, 10% are known for traditional therapy, and 1 to 5% out of this 10% have been scientifically studied and reported to possess therapeutic value. Therefore, we decided to dive into the plant and get more kind of novel plant for this particular solution. And we realized that species of Venonia are known to exhibit remarkable medicinal properties. Most of these species are also used as food. Therefore, therefore, compounds of the most predominant compound that had been studied under this particular genus Venonia is Venolis, and is currently under clinical trials, and it is reported to have anti-plasmodial activity, anti-leishmanial, anti-schistosomial, cytotoxicity, antimicrobial, and anti-inflammatory activities. Therefore, we decided to try and get a novel species of this particular genus that could help solve or contribute its quota in solving this problem. And in our search, we realized that the species Camporum is traditionally used in numerous disease treatments such as malaria, fever, hypertension, mouth sore, pains, inflammation, and skin rashes. However, from thorough scientific or literature review, we realized that no scientific data has been reported on this therapeutic use of this species. Therefore, what we are trying to solve is microbial resistance issue. That's trying to get a molecule or a compound that could be able to solve microbial issue. Also, obtaining a compound or a product that could also remove active oxygen and free radical species from the human body. Also, treat helminth infections and its resistance. Also, control inflammation by inflammation gain as a result of metabolic processes, because the current inflammatory drugs such as opioids and non-steroidal anti-inflammatory drugs, their side effect is predominantly so high. Also, because there are no scientific basis on the use of this particular plant, it's worth experimenting on it to know its efficacy. So the aim of this particular research is to obtain methanol and hexane stem bark extract of Venonia camporum We then ascertain the phytochemical screening to know the various phytochemicals that are present in the methanol and hexane stem bark extracts to evaluate the efficacy of the plant species using in vitro methods for antioxidant, the antimicrobial, anti-helminthic, and anti-inflammatory activities. Then we further separate and purify the phytochemical constituent present, and we determine the various functional groups present in the purified fraction. So in justifying our results, we know that medicinal plants have vital value to humans, and therefore it's worth experimenting on it. We also know that since the medicinal use of this particular plant is not yet reported, is worth experimenting on it and reporting its activity so that it will provide general idea to herbal practitioners and citizens of Ghana in terms of its usage. Also, it will help contribute to the improvement in health of the nation, that's Ghana and the world at large. And also, the results of the antimicrobial and antioxidant would also provide consumers and industrialists in food processing and preservation industries. So the method used in this particular research was sampling of the stem bark from Kweutafo in the eastern region, identification and authentication of the stem bark at the Department of Herbal Medicine, KNUST. We prepared the sample, pulverized it, extracted it, and conducted some biological assays on it. We performed the phytochemical screening. We did some separation and purification, and we determined the spectroscopic analysis on the purified fractions that we separated. So with the results, out of the 300 grams of the mass of the pulverized sample used, we recorded a mass of 6.317 as the yield for the extract, the methanol extract, and 0.20 as the yield for hexane extract. Also, we tested for 17 phytochemicals, and we realized out of the 17 phytochemicals, 14 of them were present in the pulverized sample and 13 were present in the methanol extract out of the pulverized sample, and 9 were present in the hexane extract. And the first 7 phytochemical is reported to be present in almost all species of Vennonia. So in determining the antioxidant assay, we employ DPPH method, which basically involves the ability of the antioxidant to be able to mob up a free radical. And the free radical we used for this particular experiment is the DPPH. And the parameter we tend to measure is the percentage scavenging, and we report it in terms of its IC50, and we realized that the IC50 of the methanol extract was 17.7 and that of the hexane was 29.54. The standard performed better than the extract. However, the methanol extract did very well in terms of scavenging free radical than the hexane. Also, we determined the ability of the extract in terms of mobbing non-radicals like hydrogen peroxide, and the mechanism involves that if you have a ferrous ammonium sulfate and you add 110 phenanthrolene to it, you're supposed to form a ferric triphenanthrolene complex. And if you have a ferrous ammonium sulfate and you add peroxide to it, it converts the ferrous ammonium sulfate into ferric ammonium sulfate, and upon addition of 110 phenanthrolene, no complex formation is observed. Therefore, if your extract has the ability to mob hydrogen peroxide, then it should be able to scavenge the peroxide upon addition of the peroxide in a solution containing the antioxidant and the ferrous ammonium sulfate, thereby nullifying the second reaction pathway. And therefore, when you add the 110 phenanthrolene, you should be able to see your ferric triphenanthrolene complex. And in this particular method assay, we observe an IC50 of 243.20 for the methanol extract and 352.20 for the hexane extract. The standard recorded an IC50 of 169.00, which means that the standard did perform better than the methanol and the hexane extract. However, the methanol extract did very well than the hexane extract. Also, we decided to determine the overall antioxidant activity of the extract, and we use the phosphomolybdenum method. And in this particular method, we prepare the phosphomolybdenum, that's the phosphomolybdenum reagent, by reacting a phosphate group with a molybdenate group in a presence of acidic condition to create a Kegen ion. And this particular reagent react with our antioxidant in 3 different antioxidant mechanism, and which include proton donation, single electron transfer, and metal chelation. And it should be able to convert your molybdenum reagent from molybdenum 6 to 5. And in this particular method, we measured the results in gram ascorbic acid equivalents per 100 grams. What it means is that if you pick 100 grams of your extract, how much of it could be able to act as ascorbic acid, which is a standard drug that we take in as antioxidant reagent or antioxidant agent, sorry. And we realized that when you pick 100 grams of our methanol extract, 31.592 could act as ascorbic acid, and that of the hexane is 30.232. So in determining the antimicrobial assay, we use the Agar-Well diffusion method, and we realized that the higher mean zone of inhibition was recorded for E. coli, with the methanol extract reporting a mean zone of inhibition of 27 millimeters, and that of the hexane 26 millimeters. The lowest mean zone of inhibition for the methanol extract was recorded for E. coli at 6 millimeters, and that of the hexane was recorded for P. originosa and B. subsolate at C millimeters. Ciprofloxacin was used as a standard drug for the bacterial, and the clotrimazole was used as the standard drug for the fungus. So the MIC of the extract were also determined. Then we realized that the MIC of the methanol extract is within the range of 0.1953 and 0.3906, and that of the extract was within the range of 12.50 to 25.50. So in determining the anti-helminthic assay, we employ the adult worm motility test, and this method basically focuses on the parameters, the paralysis time and the death time. And the smaller the paralysis time, the better the activity of the agent. And we realized that the methanol extract and the hexane extract perform better in terms of their paralysis time than the standard albendazole, with the methanol extract performing better than the hexane. The death time was also observed to have similar pattern, with the extract performing better than the standards and the methanol extract performing better than the hexane extract. So we used egg albumin denitration method to also determine the inflammatory activity of this of our extract, and we realized that the IC50 of the methanol extract was 35.83, and that of the hexane was 53.91, with the standard diclofenac recording an IC50 of 20.83. So similar pattern was also observed here, with the methanol extract performing better than the hexane extract, and the diclofenac performing better than all the extracts. So we decided to separate the various components in the extract, and we use our thin layer chromatography, and we employ 2 solvent system, which is 1:9 and 1:4 ethyl acetate is to hexane. And we realized that 4 components were separated for the methanol extract, and 6 components were separated from the hexane extract. And we therefore use column chromatography to be able to fractionalize this particular fractions from the methanol extract, because we had the methanol extract in large amounts, in large quantity than the hexane extract. So we decided collecting the 4 fractions from the methanol extract, and we collected this for fraction A, B, C, and D. So we decided to do phytochemical screening on this pure and on this separated and pure fractions, to be able to determine the type of phytochemical we've been able to separate and purify from the extract, and we realize that A, B, and C are steroids and D is a terpenoid. Therefore, we decided, okay, then let's have a rough idea about the functional groups of the pure compounds we have, and realize that from our FTI analysis or our spectrum of fraction A, we saw CHSP3 stretch, so a carbonyl stretch, CH bending, CO or CC stretch. In fraction B, we saw CHSP stretch, C carbonyl stretch, and CH bending. In fraction C, we observe CHSP stretch, carbonyl stretch, C double bond stretch, and CH bending. In our fraction D, we observe CHSP3 stretch, the carbonyl stretch, and also CH bending. And if you realize the spectra of fraction A, B, and C, because they're all steroid, they tend to have similar, how do you call it, spectra in their diagnostic region, but it was reported by Dobry. It was reported in the late 70s by Dobrina that the disparity of steroids typically lies at the fingerprint region, specifically from the 800s downwards, and similar pattern was observed here. In conclusion, we say that this experimental research has demonstrated that the extract of Venelia camporum stem bark exhibit significant antioxidant, antimicrobial, anti-helminthic, and anti-inflammatory activities. And the biological activity of the stem bark of this extract was observed to be high in the methanol extract compared to the hexane extract. And this study has provided some justification on the traditional use of the stem bark of this medicinal plant in terms of its usage in treating inflammatory disease, wounds, and skin rashes. So further work seek to separate and purify all the components in the extract. We perform some biological assays on the purified extract we collect from on the purified component we collect from the extract to determine the ones that are pharmacologically active. Then we will characterize and isolate the pure fractions that are pharmacologically active. Then we do some in vivo experimental research to be able to evaluate the pharmacologically active isolate for further experimental research. So the first phase of this work is under review at Journal of Chemistry, Hindari. These are my references. Thank you.
Okay, so thank you very much, Bernard. You also went a bit beyond your time, but I didn't stop you because we might have a bit more time. So once again, if you have any questions, please write them down, hold on. When it's time at the end, then we come in. So I said we might have a bit more time because
Hello.
I hear,
Hello.
yes. Yes.
Please, it's rather unfortunate. I'm a graduate student at Ohio University and I have a class around this time. So it's rather unfortunate I have to leave. Therefore, I will not be able to take the questions. So if someone has the question, I could address it now before I go.
Okay, so let's give that opportunity. Like I was saying, we have a bit more time because the last presenter, Samuel Jima, here is not available. So his slot will be there. So probably let me give 5 minutes for us if there's any questions regarding what Bernard has presented. If there are any questions that anyone has and would want to ask, this would be a good time. So Bernard, let me start with what is in the Q&A session. Edward is asking, what species of Vernonia genus had scientific information that got to clinical trial?
Okay, so the most studied species under Vernonia is Vernonia magdalena, which we call locally as bitter leaf, and they've separated the venolates from Vernonia magdalena and that of Venomaldalen. Yeah, so the venolates is more studied and it is in clinical trials.
Okay. And from me, so you are doing a lot of fractionations, different solvents, and all that. Is there a plan to, at some point, also look at the synergistic effect of some active fractions?
Yes, of course, because it is very possible, and that when you separate each of these fractions and you perform the various biological activities on the fractions, you might not have the exact activity as we are observing because they are synergistic effect. If you realize from the methanol extract, we have about 13 different compounds, some acting as agonists, some acting as antagonists. So based on the fractions we have and based on the structural motif we are going to get, we can decide to do some synergistic assays by combining some of the fractions to see if we can be able to get better activity. Yeah, so it's in mind.
Okay. All right. Thank you very much. Are there any other questions for Bernard before he takes his leave? I don't see any questions in the chat or in the Q&A. All right. So Bernard, we would say thank you very much for the presentation, and we will
It looks like
grant.
there's something in the chat.
Yes, the one on which species of Vernonia?
Not in the Q&A.
Okay, in the chat. Okay, Bernard, so listen to this and
Okay.
let's get an answer. So he says, since you are recommending further work on pharmacological activities, do you think your topic is still valid?
Of course, it is still valid because how do you call it? This is research, and no matter how no matter what it is, some point you might tend to have an end, but to me there is no end to research, because day in and day out there is problem coming up, and the bottom line of plant research is to be able to get active compound or the active compounds that have the activity, so that we could be able to turn it into drug. So if we are not able, since we are not yet there in getting those active compounds to be able to turn it into drug, we are still not done. And at any point in time, the results we have is reportable and publishable. So it's still relevant. It's still relevant.
Okay, Mohammed, does that answer your question? Okay, maybe.
Yes, thank you so much.
Right.
Thank you.
Okay.
Thank you.
Yes, you are welcome. So I don't see any other question for Bernard. So I would say thank you very much for the presentation and for taking the questions, and all the best in the rest of the work.
All right, thank you so much.
Yeah, all right. So we move to the next and the last presenter for the session. That's Aisha Mahama. She's with the West African Center for Cell Biology and Infectious Pathogens at the University of Ghana. And she'll be presenting work on the assessment of the immunomodulatory and toxicological effects of three anti-malarial medicinal plants in murine models. So Aisha, if you are ready and you can share your slides. Also, we would like to see your face. Okay.
Okay.
Yeah, so we can see your slides, but we can't see you.
I'm trying to show my face.
Okay.
Yes, can you see me?
Not yet, but you go on. I think what is critical is what's in your slides.
Okay. All right. Okay. Good afternoon, everyone. My name is Aisha Mahama. I'm a final year MPhil student at COBREG, University of Ghana, and the title of my project work is the assessment of immunomodulatory and toxicological effects of copper persera in
Aisha, can you check your microphone? It's your voice is going up and down. If you're using a headpiece, if you can keep it close to your mouth so that we can hear you uniformly. No, it's gotten worse. Very faintly. So it looks like you have made the situation worse. Hello?
Yes, can you hear me now?
We can hear you, but there is an echo, more or less. You go on. I think you are audible. The voice sounds a bit funny, but you are audible. So you go on.
Okay. So my supervisors are Dr. Kujua Samapusi and Dr. Samuel Leji. When pathogen invade the immune system, both pathogen interactions in the immune microenvironment determine whether or not the body remains healthy or becomes diseased. So as a result, the immune system is constantly surveying the body in order to detect and combat any invading pathogens. However, the immune system is also very susceptible to disorders, including autoimmunity and hypersensitivity. Therefore, host-directed therapies are a new branch in science that are looking at producing immunomodulatory agents that boost the immune system against a wide range of infections. And plants are a potential source of such host-directed therapies because they are more accessible and cheaper. My project work is looking at two prominent medicinal plants.
Hello, Aisha.
Hello.
Your voice is gone off again.
Okay.
Very faintly. I think a minute ago you were much better. So whatever you were doing, then go back to it.
Okay. How about now?
Getting better.
Okay. Can I continue? I should try it a couple more times.
Just try whatever you're doing. Try a bit more. Let's see.
Okay. How about now?
This is better. So please stay there. Don't move.
Okay. Thank you. Okay. So as I was saying, my project work is looking at two prominent medicinal plants, Carpa procera and Arconia cordifolia, which are widely used in Africa for the treatment of a wide range of infections, including gastrointestinal, parasitic, and recently it has also shown in vitro plus antiplasmodial activity. So the rationale behind my study is that instead of targeting pathogens, which eventually lead to a drug pressure that drives selection for antibiotic resistance, it will be prudent to also look at boosting the immune system so that you can mitigate this selection. And also, in boosting the immune system, you're going to have it competent enough to fight a wider range of infections. We've seen that there's been an increase in viral pandemics as well. So these immunomodulatory agents could serve as a potential treatment for such viral infections as well. On the other hand, however, there's been an adverse health effects that are reported with the use of medicinal plants, and therefore will be very important to look at the safety profile of the medicinal plants that will be that are being proposed to be used as immunomodulatory agents. So my aim is to assess immunomodulatory and toxicological effects of Carpa procera and Arconia cordifolia, and this will be done in murine models. And my specific objectives include the identification of markers of immune activation, conducting hematological and biochemistry analysis at acute and subacute toxicity stages, and also conducting histopathological assessments at these acute and subacute toxicity stages. So for my objective one, which is to identify the immune markers, I'm currently working on it and I'm using flow cytometry. So I moved on to my objective two, which is assessing acute toxicity study. This was a study that's conducted over 14 days, and to do this, I had 3 test groups of mice, and I administered a very high dosage of 2,000 mg per kg. And then to a control group, I administered 10% DMSO as the vehicle. Then after this one-time administration, I observed the animals over a period of 14 days, and then euthanized them on harvest major organs. Now, the most critical thing to look out for during the acute toxicity study are clinical signs of toxidromes, which could include loss of air, inability to move, inability to breathe properly, and also severe or drastic weight loss. So after this acute toxicity study, which I've been able to do for 2 of my fractions, the ACL 1:1 fraction, and then the CPL 100% fraction, I decided to compare the absolute weights of the major organs I harvested, comparing the test groups to the control groups. And what I noticed was that there were slight variations in the weight as observed in the graphs, but there were no significant changes between the test groups and the control groups. So, and then throughout the study, the animals were also very healthy. They gained weight properly, as I'll show very soon. And then for the subacute toxicity study, it was conducted over a period of 28 days. And then to do this, I had 3 test groups of mice, respectively administered 100, 300, and 1,000 mg per kg per weight of the animal. And then to the control group, I administered 10% DMSO. And I did this daily administration using an oral gavage for 28 days. The animals were euthanized at 29 days. And I also took weekly measurements of weight in order to track any weight changes that was occurring. So on the 29th day after the organ harvesting, I was also able to acquire blood, which I used for the biochemical and hematological analysis. So basically, my methodology is that to assess the toxicity at acute and subacute stages, I took blood from the animal. I went through a biochemical and hematological analysis, and the biochemical analysis was basically trying to look at liver and kidney function. And then also the hematological analysis was to look at any major blood parameters. So then the last thing I did was histopathological examination of the liver and kidney. And then with this, I fixed the organs that were harvested in 10% formalin, trimmed it into a particular orientation. And I took it through a series of dehydration processes using increasing concentrations of ethanol and then increasing concentrations of chloroform. Then I embedded and sectioned and stained with HNE stain, and finally and captured images under the microscope. So results from my subacute study for both fractions, the ACL and CP, I observed that there was a weekly, taking the weights of the animals weekly, I observed that there was an upward trend in all groups, including the control and the test. So for each group, you could see that there was a trend of increase in weight. Also same here in the CPL fraction, which could point to healthy growth of the animals. And then I went on to do the hematological profile, and this was basically looking at major parameters for both the ACL and CPL fraction. Again, I didn't observe any significant differences between the control and then the test groups. So I did the same for the biochemical profile as well, and liver and kidney functional tests. They all looked there was no major difference. So then the histopathology, I wanted to compare the control livers to the test group livers. So on the left-hand side, what you find is that in a control liver, you have mitosis ongoing, but the same mitosis was occurring in the test group livers as well. And what it points to would be regeneration, and then regeneration in the hepatocytes is a normal occurrence that happens. There was no overboard, it was within moderate levels. And then again, I wanted to see if there was any ongoing infection, and a good marker of such ongoing infection would be a high infiltration of inflammatory cells, but there was none as seen in the left side. So this is the control group, and then this was for the test group. And this was done at a magnification of 100. Then I wanted to look at the kidney as well. And structurally, I was particular about the woman's cancer because the woman's capsule is supposed to filter stuff from the blood. And so I wanted to basically see if it was a good shape. So it was a control.
Aisha, your voice is almost gone.
Hello.
Yes.
Okay. So I also wanted to look at still the same inflammatory cell inflammation, but I felt that was also important in detecting any infection or loss in function in the test group particularly. But in both the control and the test group, I didn't find any inflammatory cell infiltration. And so preliminary, I can say that both ACL and CPL fractions showed caused minimal changes to the liver and then the kidney. There was also no significant functional effects as reflected in the biochemical and hematological examinations. Ongoing work is to determine the immunomodulatory activities of these fractions, and so I'm doing some flow cytometry assays. I'm also evolving the work to look into in vivo anti-malarial properties that these fractions might show also in mice. So I'm finally finishing up with the acute and subacute toxicity study of the last fraction, ACL ethyl acetate. And my expectation is to identify a safe immunomodulatory agents that can boost immunity against a wide range of infections, including malaria. So I'd like to thank Dr. Bedore for this. I'd also like to extend my gratitude to my supervisors, Dr. Kujua Samakusi and Dr. Semoliji. I'd like to thank Dr. Kama, Dr. Ashwa Tete, all staff of Animal Experimentation, and all staff of the Knowledge Department, both at the Noguchi Memorial Institute for Medical Research. Thank you.
Okay, so thank you very much. And I hope our audience were able to at least catch what you presented. There were times that the sound was a bit bad. Anyway, so this is the end of the third presentation. So I would at this point invite questions for both Emmanuel and for Aisha. Bernard, I am sure is left. But for Emmanuel and Aisha, if there are any questions that we want to ask, if there's any clarification that is needed or comments on what has been presented, please go ahead. So you can either lift your hand, raise your hand so that you're invited, or you can leave your questions either in the Q&A window or in the chat window. So anyone with a question? All right, so let me begin by checking with Emmanuel. Emmanuel, are you on?
Yeah.
Okay, all right. So if I followed your presentation correctly, you have looked at models with individual, should I say, features. I can't, maybe I will get the terminology wrong.
Yes, yes.
But yes. So is there a way to look at multiple factors in these models, or would it be too complex to interpret?
Yeah, regarding this model was more focused on known factors. For instance, the motivation was the existence of known sites, right? So if so if you have any other feature, that will mean that there should be a modification of the model. So if you have the case of 6 features, or if you are considering other features, then you have to redesign the model. So I should say that the model is more or less feature specific.
Yeah, so my point is these 6, let's take the example of 6 features that you looked at. Did you look at them individually? So are they modeled individually or they are modeled together?
They are modeled simultaneously.
Okay.
Yeah.
Okay. All right. Yeah. Okay. Yes. So the floor is still open if there are any questions for Aisha and okay. So George, your hand is up.
Yes. Question for Emmanuel. So how is this model applicable for other viral outbreaks, such as the one we are confronted with? To what extent are any of the things that you did here translatable?
Yeah, it is possible. What will make it applicable is the identification of specific features that accompany the variants that are currently circulating. And the problem will be that we always need abundance of data in such kind of modeling. And that, I think, will be a challenge. If not that, it will be available. It will be possible to apply to the current problem we are facing.
Okay, thank you very much. So Aisha, there's a question for you in the chat window. So the question is, how might your study be affected by using animal models for malaria? How might your study be affected by using animal models for malaria? And this is from Adin.
Thanks for the question. So far, what I've observed is that for my negative control for the anti-plasmodial work, the mice have started dying. So I think that's a major challenge. They are not able to survive throughout the study. So that might be one thing. And then another thing is that even when they do survive and then you need blood for some of the downstream work, especially in the mice, their blood turns out to be insufficient because they are so tiny and then they become anemic. So yeah, you don't get enough blood.
Okay.
So those are
would it work if you switched to maybe a different parasite?
No, I think maybe I'd rather maybe probably switch to rats because they are bigger and then that might solve the problem of insufficient blood. But as to moving it to a different parasite, I'm not certain about that. I don't think.
Okay. All right. So any other questions for the two? Because if there are no other questions, then we can bring this session to a close. There will be a bit of time, so I don't know if the next session will start immediately or a little break will be okay. But I think we can bring this session to a close in the absence of further questions or comments. And I'd like to thank everyone for being with us. So my name is Kwejo Asamoah-Kwesi with the Norwegian Memorial Institute. I realized I didn't introduce myself at the beginning. All right. Okay. I thought I had another question, but it's not a question. Okay, so I will quickly hand over to Max, who will be handling the second session under this topic.
Thank you, everyone. I hope you can hear me. Okay.
Yes, we can hear you.
And final session of our conference. As usual, we have 4 speakers lined up: Jocelyn, Masood, Emmanuel, and Thomas. So do what you usually do. Get your pens and pencils, paper for the question time. So we are starting with Jocelyn from University of Ghana, Legon, who is going to speak on pesticide residues in leafy green vegetables and irrigation waters in the Accra metropolis, Ghana. So Jocelyn, you have the stage. Jocelyn, we can't hear you.
Sorry. Good afternoon, everybody. I'm Jocelyn. I'll be presenting on pesticide residues in leafy green vegetables and irrigation waters in the Accra metropolis, Ghana. So different types of vegetables are produced in Ghana for food, but it's been observed that these vegetable farming is normally associated with several pests and diseases, which usually affect the yield of crops. So in order to control this pests and diseases and associated crop losses, different types of pesticides are employed. Normally the organochlorines, the organophosphate, and synthetic pyrethroids are what the farmers are normally observed to use. The organochlorines are normally chlorinated hydrocarbons, which are neurotoxic in nature. They accumulate and they are not easily biodegradable. Because of that, they've been banned in several countries, including Ghana. The organophosphates are also organic compounds with central phosphate groups. They are also neurotoxic in nature, normally attacking the enzyme acetylcholine esterase, thereby killing the pest, but they also can affect humans based on the concentrations used. And then the third type that we normally observe the farmers using are the synthetic pyrethroids, but they are synthesized from a flower. So they normally don't persist in nature for long. They are easily degraded by the presence of the heat of the sun. So you observe the farmers using some of these pesticides in controlling of their vegetables. Although these pesticides are found to be effective, but we observe that normally the farmers tend to use either more of these pesticides or they don't obey the manufacturer's instruction, which normally leads to increased concentrations of pesticide residues in the vegetable samples. And these pesticide residues can persist in the vegetables for a long time and can even be ingested by consumers, and we don't know what its potential effect in consumers. And it can also persist in the soil and pollute neighboring water bodies. Some studies have shown that the presence of this pesticide in foods or contamination of humans by these pesticides are linked to some cancer, some birth defects. So because of this study looked at determined the presence of pesticide residues in leafy green vegetables cultivated in vegetable farms in Accra metropolis, And then we also looked at the irrigation waters, which are used by the farmers to ascertain the safety of this produce that are consumed by residents in Accra and Ghana at large. So for our study, we visited farming areas in different parts of the Accra metropolis, but we had 2 outside which we were willing to participate, so we included them. So usually we visit a farming community, and the farmers normally tend to work in like a form of a community. So the red marks we see on the map are the farming areas that we visited. So any farming area can consist about between 5 to about 40 farmers who are independently working, but they work in a community form. So we visit the farming area, we let the farmers, their heads get to know what we are doing, and then those who are willing to participate, we include them. So we visited these farming areas, we sample, if a farmer has a produce, any of the vegetables of interest to the study, and they are ready for harvesting, we buy some, then we transport them to our labs. So for the irrigation water, this is an example of a waste drain. So normally with the waste drain, it's a waste drain that is passing by the farming area that the farmers tap into. So this farmer was actually fetching this water to use on his produce. And this is an example of what we termed as a river or a drain. It's actually a river that the farmers claim it comes from the mountains in the Brie and passing through Accra, but because of human activities around, it's beginning to look like a drain. That is why we put in quotes a drain. And then we have the third type of irrigation water, which are the shallow wells. These ones, the farmers normally dig them themselves, and then they will either pump water into it to use closer to their farms, or they rely on rains to fill it up. So for the kind of vegetables that we used the 4 types, which we refer to as indigenous Ghanaian vegetables. So mainly the amaranthus species, or commonly called alefu, the Cocros olitorius or ayoyo, the hibiscus sabdafari, which we normally call ashure, and then the Solanum agrocarpon, which is commonly called buma. And then we use 2 other vegetables we claim as exotic to the Ghanaian vegetable farm, and that was the lettuce and then the cabbage. So when we visit the farms, in total, we were able to collect about 34 vegetable samples. And then these were from 15 different farmers in 10 different farming areas. And then we sampled irrigation water too from 10 farming areas. And these waters were served over 40 farmers in the areas that we sampled from. So these, we collected these samples, and then we transported them to the pesticide residues lab of the Ghana Standards Authority. So in the lab, we added aceto, we commute the vegetables, and then we added acetonitrile and other salts and buffers such as sodium chloride, magnesium sulfate, and hydrous, basically to extract the pesticide residues that may be present in the samples. And then we clean it up with the primary, secondary amide and magnesium sulfate so as to reduce interferences with our results. And then we use the GC equipped with the pulse flame photometric detector and a mass spectrophotometer to detect the pesticide residues. So with the pesticide residues, those of concerns were organochlorines, organophosphate, and synthetic pyrethroids. But basically the ones that the Ghana Standard Authority has been monitoring for them in their presence in produce that are consumed in Ghana or exported. So those were the ones that we also use for our study. So for the organochlorines, examples of them were DDT, lidane, aldrin, heptachlor. And then for the organophosphate, chlorpyrifos, diazinon, melathion. And then for the synthetic pyrethroids too, examples of what we looked out for was aletrin, cypermethrin, and the lambda cyhalothrin. So after I prepared the different concentrations of the standard solutions, and then we used it to prepare standard curve that helped us to estimate the presence of these pesticides in our produce or our samples. We observed that for the vegetable samples, about 50% of them, as in 17 out of the 34 samples, had pesticide residues in them. And then for the irrigation waters too, about 52% of them had pesticide residues in them. But the good thing is that we did not observe any organochlorine pesticide in any of the leafy vegetables or irrigation water. This could be because its usage has been banned in Ghana for over 2 decades. Or it could also be that they may be used, but maybe in very small quantities. That is why we didn't detect. But we just hope that our first explanation will be what is actually happening, that the farmers are not using it. But actually, in interacting with them, because this is part of a bigger study, none of them mentioned DDT. It was just one old farmer who mentioned DDT. But when I asked their head, he was like, for the aged ones, every chemical, every pesticide they refer to as DDT. But I analyzed that farmers produce and I didn't find any DDT inside. So I will assume that they just refer to any pesticide as DDT. And then we also observed that 9 of the leafy vegetables had pesticide residues exceeding the adopted EU maximum residual limit. I'll talk a little bit about that later. In the Ghana Standards Authority, they were using the EU MRL, mainly because most of these produce are exported to the European markets. So for the analysis of the irrigation waters, we observed that chlorpyrifos, which is a form of organophosphate, and permethrin, which is a form of synthetic pyrethroids. Those were the 2 main pesticide residues that were found in the irrigation water. 3 out of the 4 river or drain water samples had chlorpyrifos in them. 3 out of the 11 shallow well waters had chlorpyrifos in them. And then 6 out of the 8 waste drains that we analyzed had chlorpyrifos, and then 2 of them had the permethrin in them. With the leafy vegetables, we observed different kinds of pesticide residues in them, both from the organophosphates and then the synthetic pyrethroids. So for the amaranthus species, for instance, which we locally call alefu, out of the 8 samples, 3 of them had deltametrin in it. For the cabbage samples, we had 3 that we analyzed. Only one of them had pesticide residues, but this particular samples had 5 different pesticide residues in them. This could be because interacting with the farmers, they claim it's very difficult to grow cabbage without pesticides. So they tend to use a lot of pesticides on them. We had some farmers who claimed to apply pesticides over 8 times in the planting cycle of a cabbage. So it could be that this particular sample could be for such a prospective, such a farmer. That is why we got a lot of the pesticide residues showing in them. Out of the 4 C. oletorius samples, 3 of them have various pesticides in them, mainly being the chlorpyrifos, cypermethrin, and then lambda cyhalothrin. With what we locally call the shree, we had 2 samples. Another 2 samples had pesticide residues in it, namely the chlorpyrifos and the lambda cyhalothrin. With lettuce, we had a lot of lettuce samples. We had 14 in total, but 6 of them have pesticides residue, but mainly being the chlorpyrifos, and then the cypermethrin. And then what they locally refer to as the BUMA or the S macrocarpon, we had 3 samples, and then 2 of them had various pesticide residues in it. And then we also observed that among the, so out of the 70 samples that had the pesticide residues, as I said earlier, for 9 of them, the residues were above the adopted EU maximum residual limit. And these pesticides were mainly the chlorpyrifos, the lambda cyhalothrin, and then the deltametrin. And if you look at that, so I show different colors for the different pesticides, you could see that the chlorpyrifos, which is this shade, we saw it being used by most of the vegetables, and then present in most of the vegetables, sorry. And then this shade is the deltametrin, and then this color is the lambda cyhalothrin. So those were the 3 pesticides that some of them were exceeding the EU adopted limit. And these results too were not too different from other studies that have been done in the region, of also reporting about 50% and mainly chlorpyrifos for controlling pests and diseases. Interacting with the farmers and then them giving us some of the names of chemicals they use, realized that most of the chemicals that they mentioned had chlorpyrifos or lambda cyhalothrin. So it may not be too surprising that these 2 pesticides were seen a lot in the vegetables that we sampled. But it's also scary that these consuming vegetables with all these kind of pesticides residues in them, there could be a potential that consumers could see some side effects which may not have been looked into. So in conclusion, about 50% of our vegetable samples and irrigation waters had pesticide residues in them, with about 26% of the vegetables having the residues exceeding the EU MRL. And with this information, I think the farmers, they are aware. Some of them are aware, talking with them, some of them seem to be aware, but I think we need to encourage them more about the effect of these pesticide residues in produce, so they should stop abusing it. And then we should just encourage them. They're interacting with them, I realize that they always feel like you're coming to arrest them, so they try to hide what they are doing. But I think if we encourage them and let them know that this is not good, but do it the right way, I think maybe in the long run, it would help reduce the presence of pesticide residues in vegetables, which are commonly grown and consumed in Accra. Thank you.
Thank you, Joycelyn, for a beautiful presentation. So if you have any questions, hold on to them for now till it is question time. We now have Masood Suleiman Teller. Masood Suleiman Teller from the Department of Pharmacology and Pharmaceutical Sciences from the Ahmadu Bello University of Zaria. He is presenting on the effects of co-administration of piroxicam and methanol stem bark extract of Berkia africana L on acetic acid-induced breathing test in mice. So Masood, take over. Masood Teller, you have the stage. Masood Teller. We are not hearing from him. I don't know what is happening.
Well, yesterday he was on, but today I've not seen him. So let's go on.
Let's go on then. Emmanuel Korti, are you with us? Emmanuel Korti from the Department of Chemistry.
Yes, please.
Thank you. From the Department of Chemistry, College of Science, Kwame Nkrumah University of Science and Technology, Kumasi, to present on the investigation of some pharmacological activities of Maria Mercanta leaves. So Emmanuel, take over.
Okay. Thank you very much.
Your slides, but we can see.
Okay.
Good.
So I hope you can see me right.
Yes, we can see you now.
Okay.
Go on.
I'm Emmanuel Korti. Sorry, please, I'm coming. Good afternoon. I'm Emmanuel Korti, working with Mod Adole Akwe. We are supervised by Professor Akwesi Echampo at the Department of Chemistry, KNUST. I'll give a talk on what we've been doing in our lab, which is investigation of some pharmacological activities of Maria Mercanta leaves. I'll talk about the introduction the problem statement, aims and objectives, methodology, results and discussion, conclusion, and some references. Since ancient time, plants have been used in the treatment of many ailments. They are believed to contain secondary metabolites that are responsible for these actions. Besides it being readily available, they are very effective and economical. And the major disadvantage, however, is the fact that they have multi-therapeutic effects. Among these known plants is the plant Maria mecantha, which is used for medicinal purpose, for treatment of waist pain, treatment of worm infection, and gastrointestinal disorders. According to WHO, 70 to 95% of the population in the developing country relies on plants for their primary health care. The improper intake and disposal of anti-inflammatory and antioxidants, antimicrobial have been a factor for the development of resistance of pathogens towards antimicrobial. So this has been a problem that is faced by the world at large. And also, new compounds of higher potency and efficacy is needed to replace less potent and less efficacious anti-inflammatory, antioxidant, and anthelmintics. And lastly, the plant itself is used in the treatment of anti-inflammatory, that is for anti-inflammatory, antioxidant, antimicrobial, and anthelmintic properties. These uses have not been scientifically proven. So it is therefore justifiable for us to embark on a research to search for a novel compound for novel drugs, and also to scientifically prove that indeed the plant Maria mecantha is used for these properties. So we therefore seek to investigate some pharmacological activities of methanol and acetone extracts in Maria mecantha leaves by one, to extract components from the leaves of Maria mecantha using methanol and acetone by means of sourcelet apparatus, to screen the phytoconstituents that are present in the crude sample and crude extracts, to investigate for the anti-inflammatory, antioxidant, antimicrobial, and anthelmintic properties in the methanol and acetone extract. And lastly, to determine the number of components and functionalities present in the extract by thin layer chromatography and FTIR analysis. So we therefore sampled the plant at Obuasi, Kumasi, dried and milled it, did sourcelet extraction using methanol and acetone as solvent, ran some phytoconstituents on the crude extract, and also bioactivities on the crude extract, that is anti-inflammatory, anthelmintic, antimicrobial, and antioxidants. And lastly, did FTIR analysis and thin layer chromatography on the crude extracts. So from the results, 12 phytoconstituents were screened on the crude extract and methanol and acetone extract, of which the crude sample and methanol extract contain all the 12 phytoconstituents, whilst acetone contain only 7. This as a result of the polarity. Acetone extract is less polar. So in view of that, it couldn't abstract the highly polar constituents. These phytoconstituents are responsible for the actions. Blood dilution assay was employed to test for the antimicrobial ability of the extract, since it is used in the treatment of gastrointestinal disorders, which are caused by microbes. The microbes used were E. faecalis, S. aureus, S. typhi, P. aeruginosa, K. pneumonia. The minimum inhibition concentration was determined for both extract and the standard drug, of which the standard drug was ciprofloxacin. The highest activity observed with the use of methanol extract was against S. typhi and S. aureus with MIC of 1.56 milligram per ml, whilst that of the acetone was 1.56 milligram per ml, and that was for E. faecalis. And also for the standard drug, it was E. faecalis. The phytoconstituents that are present that may be responsible for this action are flavonoids, phenols, and tannins. In vitro anthelmintic assay was also employed in this study to determine the anthelmintic potency of the extract by using earthworm. In this assay, the standard drug was mebendazole, and it was tested against the concentration of 0.5 milligram per ml and 0.031 milligram per ml. The paralysis time and death time was recorded, of which acetone extract gave us the lowest paralysis time and death time, followed by methanol extract and also the standard drug mebendazole. The lower the time, the more potency the extracts. And also the p-value for the hermetic assay was less than 0.001. That is in comparison of the two extracts against mebendazole. The phytoconstituents that may be responsible for these actions are flavonoids and tannins. Antioxidant assay were employed using three methods, that is DPPH, H2O2, and TAG. With DPPH radical scavenging assay, the IC50 was calculated for both extract and the standard drug, that is ascorbic acid. This is to test for the ability of the extract to mob the free radicals that are found in animals and humans. Concentration range between 6.25 microgram per ml and 100 microgram per ml were tested. It is shown from the results, it is shown that methanol extract showed the most potency of IC50 4.5 microgram per ml, followed by ascorbic acid 5.38 and acetone extract 20.78, which means that methanol extract is of highly polar than acetone extract. H2O2 assay was also employed to test for the ability of the extract to mob the non-free radicals that are present in the extract. Here, the IC50 was determined, and also methanol extract gave us the lowest IC50, followed by the gallic acid, followed by acetone extract. The total antioxidant capacity of the antioxidant assay were employed using phosphate molybdate assay. In this assay, we are looking for the capacity of the extract that will perform as ascorbic acid. Here, a graph of abundance of ascorbic acid were determined by using the equation to calculate the total antioxidant as expressed as gram ascorbic acid in 100 grams. From here, we can see that methanol extract gave us 99.75 GAAG, and acetone extract gave us 93.10 GAAE. The higher the value, the more potent the extract. So it means that methanol extract will be more potent than acetone extract. In comparison of all the antioxidant assay, methanol extract proved to be more potency than acetone extract. With in vitro anthelmintic assay, the potency of the plant were also employed using in vitro egg albumin denaturation, since the plant is used for the treatment of waist pain. Here, the standard drug used was aspirin, and the percentage inhibition of both extract and aspirin were determined at the concentration of 1,000 microgram per ml to 5,000 microgram per ml. At concentration of 5,000 microgram per ml, it is observed that aspirin gave the highest inhibition, followed by acetone extract, and then followed by followed by methanol extract. In comparison, at 5% p-value, it was significant. FTIR analysis of the food extract was also determined to know the functionalities that are present in the extract which are responsible for this action. From the FTIR analysis, we can see that both methanol extract and acetone extract give us similar functionalities, of which OH was around 3,247, CH-sp3 was around 2,900, C double bond O was around 1,700, and then alkene CC double bond was around 1,600. Thin layer chromatography was also employed to know the number of components that are present in the extract. From the result obtained, hexane against acetone gave us the best separation solvent, that is 8:2. Hexane was 8 and acetone was 2 of RF values between 0.24 and 0.60. We can see from the visible that these are our A, these are our B, these are our C, and these are our D, which shows a wide separation between them. In conclusion, we can say that from the result obtained, it confirms that indeed the plant is used in curing those diseases. And also the acetone extract has anti-inflammatory and anthelmintic potency than methanol extract, whilst methanol extract proved otherwise in anthelmintic assay. Both extracts showed remarkable antimicrobial potency. 4 components were separated from the extract using TLC. Flavonoids, tannins, steroids, phenols, polyphenols, phytosteroids, quinones were present in both extracts, except alkaloid, saponin, terpenoids, anthraquinoids, and glycoside, which were present only in methanol extract. These are some of my references. I would like to acknowledge my supervisor, Professor Akwesi Echampo, and also Mr. Abdullah Nazo, that is MPhil student, for their support, and also my partner, Mod Adole Akwe. Thank you.
Thank you, Emmanuel. As the tradition goes, line up your questions. At question time, we'll deal with them. Now we have Thomas Intebi. Thomas, are you with us? Thomas Intebi, Department of Chemistry, KNUST. Thomas Intebi is to present on in vitro antimicrobial, antioxidant, anti-helmintic activities and TLC analysis of Kligelia. Thomas Intebi. Thomas, you need to unmute and share your screen. Thomas, if you can hear me, please unmute and share your screen. Okay, so can you hear me, Thomas? Can you hear me, Thomas? Yes. Thomas is connecting to audio. Good. So now you can share your screen and start your presentation.
Thomas, did
Thomas
you
is from the
enter?
Department of Chemistry, KNUST. Yes.
Hello.
Yeah, Thomas, did you enter as a panelist or as an attendee? If you entered as a panelist, go to share screen at the bottom and share your slides.
Please, I entered as a panelist. I'm trying to get.
Okay, good.
We are now waiting for Thomas Interby of the Department of Chemistry to speak on in vitro antimicrobial, antioxidant, antihelmintic activities and TLC analysis of Kigelia angulensis. So Thomas, if you are ready, we are waiting. Good, we can now hear you. So you can share your screen and begin your presentation. Good. Can you see the green button at the bottom of your screen? Thomas. We are still waiting on Thomas. Thomas, you are the next speaker. We are waiting on you.
Hello.
Nana Pepper, if you have Thomas' backup slides, can you put it on for him? Okay, he started.
Hello.
Thomas is still trying to connect and share. I think we're almost there. Okay, so whilst waiting for Thomas, probably good. We haven't seen your slides yet. Can you find your slides? Hello, Thomas. Good. So now go to presentation mode and share from your screen. Thank you, Thomas. Unmute. I think you are muted. Unmute. The microphone is muted, so unmute. Cannot hear you at all. Still cannot hear you.
Thomas, perhaps you should check your volume or remove your earpiece. It might work without the earpiece.
Thomas, check your volume because we still cannot hear you. Thomas, we're still waiting.
Yeah, George, I agree with you. Maybe we should move to question and answers whilst he sorts himself out.
Okay. So do we have any questions? Joyce Ling, are you with us? And Emmanuel Corte? Joyce Ling and Emmanuel Corte.
I'm still here. I'm still
Good.
here.
Yes, please. I'm still here.
So do we have any questions on the floor?
Yeah, my hand is up.
Okay, I'm reading from F and Sam.
My hand is up.
Okay, Akwesi, go on.
Okay, this question is for Joycelyn.
Okay.
You tested for certain pesticide.
Yes, please.
I'm sure there could be more which you didn't test for. So I want to know why you restricted yourselves to only these pesticides. And then, did you do any antimicrobial? Because apart from pesticides, looking at the water you showed us, it could be deadly. And then what would be your recommendation, especially with lettuce, because that's what we normally eat raw. Cabbage, a lot of people cook it, but lettuce, most of the time is raw. So what would be your recommendation with lettuce, looking at the amount of pesticides in the lettuce you showed us? Thank you.
Okay, thank you. So I chose these pesticides because those are the ones that were being monitored by Ghana Standards Authority. These are the ones they routinely monitor for in fresh produce which are consumed or exported from Ghana. That is why I focused on those pesticides. And then whether I did microbial, yes, I did microbial. And then the results was also not too good. We had fecal coliforms of about 6 log, and we had salmonella, I think salmonella in about 10% of the vegetable samples. And then we had the indicator microorganisms are pretty high. We had salmonella in some of them. So I would say the microbial results were not too good. I think the pesticide results are a bit better than the microbial results. And then my recommendation is that because these are not consumed, consumers should, the sad aspect is that the alternatives may not be too good in the sense that normally we advise the consumers to wash this produce well with some sanitizers before we consume. But for something like microbial count, it can lead to a reduction, but with pesticide residues, it cannot. So we just have to be more, work more with the farmers and help them to do the right things so that they can reduce these pesticides or microorganisms associated with this fresh produce that we consume. Thank you.
Is a question answered?
Yes, I have to accept it. I mean, she can't do everything.
Okay. Rekubano, I saw your hand up. Rekubano, I saw your hand up.
Okay, good afternoon. Please, I wanted to ask Joycelyn, the pesticides that she found, I wanted to know the concentrations and then if the concentration met the EU standard as she wanted to do. And then to Kwete, I wanted to find out why he didn't do the phytochemical analysis on the plants, but on the plant extracts instead.
Okay, so with the pesticide residues, I don't have the values offhand, but we observed that some of them were higher compared to the EU MRL. They were, in terms of the high, most of them was not, it was less than, I think it was between 30% higher than it. It was only one that was twice the MRL value. There was only one pesticide residue that I observed that it was more than double in vegetables, but the rest were about 30, maximum about 30% higher than the MRL.
Rekubano, is your question answered?
Okay. Yes, please. And then to Mr. Kwete, why he did the phytochemical analysis on the plant extract and then not the raw plant. And then why did he opt to use aspirin as a control for the anti-inflammatory effect and not any other anti-inflammatory drugs like diclofenac or brufen? Thank you.
Okay. Thank you for the question. With the first question, I did phytochemical screening on the crude sample. During my presentation, I stated that 12 phytoconstituents were present in both crude sample and methanol extract. So indeed, I also ran or screened the phytoconstituent on the crude sample before the crude extract. That answers the first question. So with the second question, the standard drugs that we use, we are following a protocol, and per the protocol, they said that we can use any of them. So with respect to the cost, that is why we opted aspirin to ibuprofen and the rest. Thank you. I hope it answered.
Yes, thank you.
You're welcome.
So, Abdul Fattah, your hand is up. Abdul Fattah, do you have a question?
I have a question for Emmanuel Cotti.
Good. Go on.
Yes, Emmanuel Cotti, I want to find out from all the biological activities that you've performed, you realize that the methanol extract performed better than the acetone extract. Can you get us a reason for that? Yes, that's my question.
Okay. Thank you very much for your question, Abdul. You can see from the phytoconstituent that methanol extracts contain most of the phytoconstituent as compared to acetone extracts. So it can be as a result of methanol being more highly polar than acetone extract. That is why methanol extract perform more than acetone extracts.
Is your question answered, Abdul?
Yes, it's answered. Thank you very much for that.
Okay. I think for now, would like to move on. Majid. Abdul Majid, are you with us? Abdul Majid, are you with us? Okay, the questions in our chat box. Please, can you give us a literature perspective on the long-term effect of these pesticide residues on the human body? And this is to Joycelyn.
Thank you. With the organochlorines, which have been banned for years, there are some studies that claim they are carcinogens, although there's a dispute on it, but there are some studies that claim they are possible carcinogens. But with the organophosphate and synthetic pyrethroids, normally they just lead to some effect as in headache, muscle weakness. Those are the kind of side effects which have been reported in literature. But it's the organochlorines that normally they seem to be more deleterious. Let me put it that way.
There's a question about EU standards from Rekubano again. What are the concentrations of these pesticides found, and did they meet the EU standards?
So I think I answered it that so if I don't have the individual values in mind, but in general, it was so it's 9 out of the 17 that had their values being higher than the EU MRL. So the remaining 8 were below the EU MRL. But it was only one sample that the concentration was double, but the rest it was about 30% more than the EU MRL values. MRL, sorry.
So Fred, your hand is up. Thank you. Fred Aiti. Fred, your hand is up. Okay. Do we have Abdul Majid with us?
I think there are 2 more questions from F answer.
Okay.
If you go to the question box to Joycelyn.
Okay. So he asks about association between pesticides and pesticide residues and cancers. That's why I said that it's with the organochlorines that there are some studies that shows that they are possible carcinogens, but not with that. I've not seen anyone with the organophosphates or the synthetic pyrethroids, maybe because those ones are not bioaccumulative or they easily disintegrate as compared to the organochlorines. And then with the short-term effects of this, so with the chlorpyrifos, it's normally headache, nausea, dizziness, and some of the farmers that say to me that after using the pesticide, after applying it, they begin to experience headache and nausea, maybe because of their exposure, current exposure, and them not wearing the appropriate attire. Those are some of the side effects you find in literature. Now with the lambda cylohydrin, it's kind of like you have burning sensations or prickling feelings on your body, which some of the farmers also do claimed during the study that that's what they feel after applying the pesticide. So those are the short term, but with the long term one, I I've not seen any much information about the long-term effects aside these short-term observations that the farmers who apply them normally experience when they do.
And is there any link between these pesticides and cancer?
With the organochlorines, as in the ones that I saw, or the ones which were exceeding the MRL, I haven't seen any study so far claiming they are potential carcinogens. The potential carcinogens are normally with organochlorines, and that's the one that I said I didn't find any so far in the vegetable samples or the water samples that we analyzed.
Very much. Thank you, Joycelyn. Fred, IAT, your hand is up. Fred, IAT, your hand is up. You have a waiting on you, Fred. Okay, whilst waiting on Fred, Abdul Majid, are you with us? Abdul Majid is replacing Masood Suleiman. So Abdul Majid.
Yes, sir, I'm the last. Yes, sir, I'm ready. I'm the last.
You have an echo in the background. Okay. What about Thomas Ntebi?
Hello, sir. I'm ready. I'm the last.
Okay. Share your screen.
All right, sir.
Speaking on the effects of co-administration of piroxicam and methanol stem back extract of Beckia africana. He is from the Department of Pharmacology and Therapeutics, Faculty of Medical Sciences, Ahmadu Bello University in Zaria. Abdul Majid, we're waiting on you. Abdul Majid.
Okay, sir. I'm coming, sir.
Emmanuel Korti, this question is from Fred IAT. Emmanuel Korti, are you with us?
I'm trying to share the work.
Whilst we wait on Abdul Majid, Emmanuel, this is your question. Can you explain how protein denaturation contributes to inflammation, and how possible can your extract prevent the denaturation and hence its anti-inflammatory effects? Emmanuel.
Okay, sir. Let me share the work.
So there's a green button at the bottom of your screen, the share screen. Hello, Thomas. Thomas in Tebi.
Hello, please. The question again.
Okay. The question says, and I read, can you explain how protein denaturation contributes to inflammation, and how possible can your extract prevent the denaturation and hence its anti-inflammatory effects? Did you get it?
Yes, please, I do.
Good. How can
Thank
your
you very much.
Go on.
Okay. Thank you very much for the question. So protein denaturation actually is a form of inflammation, and this inflammation, we can prevent it by using, say, aspirin. So when aspirin is attacked on the inflammation, it reduces it. So here we are looking for the ability of the ability, we are looking for the ability of the extract if it is able to reduce inflammation. That's what we are looking out for. So from my work, we can see that aspirin, acetone extract, and methanol extract was of the same, almost the same percentage inhibition at concentration of 5,000 microgram per ml. So this can be justifiable by saying that since methanol and acetone extracts are of the same as aspirin, that is a standard drug, it means that it can also serve the same purpose as aspirin. And also the p-value for the in vitro anti-inflammatory assay was less than 0.01. That is 5%. Please, is the question answered?
Fred. Hello, Fred. Okay. Akwesi. Okay,
Yes. Yes.
I think our presenters don't seem to be ready and we've exhausted our questions.
Yeah, I was just checking the schedules and we are supposed to end at 4:20.
Okay.
So let's keep it open for another 5 minutes for any more questions. So this question can be to all the presenters today if they are on. Another 5 minutes. If not, then we'll just have the closing ceremony.
So we have an open session for about 5 minutes.
Yeah, for any question that anybody wants to ask.
Yes. So if anybody has any questions, this is the time to ask. An open floor. Okay, my question goes to Joycelyn. Joycelyn, how do you treat, I like lettuce a lot. So my question is, with this high concentration of pesticides, how would you treat the lettuce for consumption?
Unfortunately, the use of water or the sanitizers that we normally apply on food is not able to reduce pesticide residues. It's good for microbial count reduction, but not pesticide residues.
So what do we do?
So it has to be from the farmers, from the planting. They have to abide by the waiting period. They have to use the right concentrations. Actually, I even witnessed one farmer who had applied pesticides. I wanted to buy some and he was like, no, it's not. He has to wait for the withdrawal period before he can sell. But right before my eyes, a market woman came giving him pressure and she bought it and carried these to the market. I don't know the specific market she was taking it to. So I think it's the sellers and then the cultivators who either the enforcers, we have to encourage them to do the right thing. And those who enforce the law to must go around and make sure that they are doing the right thing. That's the only way out, because once the contamination happens, it's very difficult for you to reduce them.
Thank you very much.
Welcome.
Abdul, are you ready?
So, Max, I have another follow-up question for Jocelyn.
Go ahead.
So, Jocelyn, is there a way to even detect this? And if there isn't, I am proposing we find a way, because for the ordinary consumer, if you go to the market and you pick anything, if there's no way of knowing, you would not know how much of these pesticides is in whatever you've bought and are taken home. So is there any advancement in, for example, doing something that would quickly tell you that this has pesticide levels probably beyond what you should be consuming? If there isn't anything, is it possible to develop something like we do for diseases, having a rapid test?
Yeah, so thank you. I think, yeah, for the rapid test, maybe I have to talk with other scientists to come up with it. But one of my professors always says that the produce that you could see the insect bite, that is an indication that there are less pesticide residues in them.
Okay.
So the cabbages that have the pesticides, kind of, you see the holes rather, they rather an indication that they may be very low in pesticide residues as compared to those that look very good.
Okay. Unfortunately, they are the ones that you go to the market and want to avoid because you think
Yes,
some insects are
unfortunately, that's
putting a
it.
bit of it. Okay.
Yeah.
At least a
Okay,
good
Jocelyn. Sorry. Okay, Jocelyn.
Yes,
I'm also interested in the microbial aspects.
Okay.
So how do we wash it? Do you have any way
How
of
do we
washing
wash
it to be sure that water will not help? To be sure that we have removed a lot of microbes, since we eat the lettuce raw most of the time.
Okay, so in another study, we use different sanitizers that are commonly used in Ghana and those which are also used by fresh produce industry in the US. And I'm happy to say that with the use of lime water and vinegar, they seem to be very comparable to what the ozonated water, the chlorine water gave us. So I normally encourage people to use vinegar, wash their produce with vinegar or with lime water. Just that the lime water, how much lime you have to squeeze in order to get enough. So I think vinegar is the most convenient one that you have to wash your produce with, and then with some water. But the salt water that we normally do, it was able to achieve less than 1 log reduction, which was similar to washing it with just water. So I just recommend that we have to wash the vegetables. And then if you can add a sanitizer like vinegar, I think that would be very good.
Are you talking about apple cider vinegar or vinegar?
Yes, please.
Or there's vinegar, apple cider vinegar?
Apple cider vinegar also works. And then the normal vinegar, the plain one too, also does.
Are you talking about, say, 1 in 2?
Okay, I think we did 30%.
30%, okay.
Yes, please.
Okay.
Just that with time, it also impacts a kind of odor on the vegetables. So when we did sensory analysis, that's some of the problems some of the consumers had.
Can the person stop sharing? I want to share, but someone is sharing.
Thomas and Tebi, we are out of time now. So we are very sorry we cannot see your slides. I think we have to end because we agreed to end at 4:20 and 15 minutes will take us too much. I'm sure today is Friday, people may have scheduled their times to match with the meeting. So I'm very sorry you cannot present now. That's why I sent a message to every presenter that the system will be open 1 hour before time. So that was the time for you to try out your slides and everything, so that when it's your turn, you just don't waste time. So Thomas, we are very sorry you can't present today. The same goes for the guy at Ahmadu Bello University. Yesterday, I actually told your boss that we open at 12 o'clock, so you should present. And I'm surprised you are now even attempting to do it. And there's so much echo on your side, so yours will not even be accepted. Okay, George, do you agree?
Right.
So are you joining again for them?
Today is the last day.
Some of these things, you see what I was telling you, that come and try the thing ahead of time.
All right, ladies and gentlemen.
Right, over to you, George.
Thank you very much. I think we've had 2 days of very stimulating presentations. And I want to, on behalf of the organization, thank all presenters, all who participated in a variety of ways. We thank you for your contributions. I'm hoping that people have struck possible potential collaborations, somebody you might approach for mentorship, things that could enhance your particular line of work. So we look forward to next year. We are hoping that next time we can meet on one of the campuses in Ghana. This basically went up without much of a hitch until the last 2 presentations. So we're grateful. But on behalf of the organization, I would like to say once more that thank you so much all. Your leadership will organize additional web-based, online-based events between now and then next time we can do a workshop or an actual in-person conference. So thank you all. We haven't had an election since Kumasi, but because of the exigencies of the events over the last couple of years, the board decided to wait until we can meet in person to do a proper election for the next board. So that's why we are in holding pattern. I will, in the meantime, encourage you to consider joining if you are not a member. If you are a member, please consider keeping up with your dues. Yesterday, we put up the information, and I am hoping that you make that modest contribution towards all this effort. And I thank you. I think we are adjourned until. Oh no, I'll round up. Oh, okay. Sorry. I hand over to the chair of the scientific committee then. Yeah. So thank you all for being with us. At least today we had quite a good number. I counted up to 45 people. Compared to yesterday, it was good. And thank you also presenters for keeping the 15-minute time. That's very good. So we hope it will be like that. Next year, if it happens that we have to meet in person, UCC still has the mandate to host. We've not changed that yet. So next year, we hope that COVID will allow us to have it at UCC. And then in case anybody wants somebody else's slides
or wants to ask questions in private to any of the presenters, then you can send your mail to me and I will seek permission, or I will refer you to the person. I will send the person's email to him so that you can talk between yourselves. And if that person agrees to give your slides, it can be behind the scenes. COBREG, as a rule, doesn't send slides on request. The owner has to send it to you. And as George said, if there's somebody that you felt you wanted to collaborate with, but you couldn't get a chance to connect, you can always let me know. I have the emails of every participant and every attendee. So you can let me know and I'll also connect you to the person. I think it's been very good these past 2 days, except the last two speakers, whom I'm sure would next time try and load or try out your slides always before the presentation, whether it's virtual or whether it's live. You always have to try out your slides before the presentation. So without much ado, if nobody has anything more to say, if you have something more to say, you can just raise your hand.
There's a request.
I see. Asamokusi, is your hand up from the question or your hand is up for now?
No, it was from the question. Sorry.
Okay, so can you lower your hand?
There's a request for the dues information to be emailed to attendees. So we'll follow up.
Okay, then send it to me and then I will email it to all attendees. If anybody has any comment, just raise your hand, or then I can see something new in the chat. Okay, I don't think there's anything new. Yeah. So if there are no other questions, I think we can call it a meeting. And thank you all. And we hope you respond to the call again next year. And we hope this time we would have resolved the COVID crisis and then we can actually meet in person, hopefully on UCC campus. Thank you all.
Thank you for a good job done.
Thank you for a good job done too.
Yeah. Bye-bye. Thank you all.
Bye.